So, hi to all. I would like to welcome you to today's keynote talk of Professor Stephen Williams. So, Professor Williams got his Bachelor of Science and PhD from University of Wales. And up to 2010, he was a tenure program leader at the Emcee Laboratory of Molecular Biology in Cambridge. And now he's a professor at Queensland Brain Institute. So broadly speaking, he's interested how synaptic inputs are integrated in the dendrites of single neurons, how these input signals are transmitted and transformed while they're transmitted from the dendrites towards the soma, and how they are encoded in the action potentials of the neurons. So Professor Williams, the stage is yours, and looking forward to your talk.
Thank you. Hello, welcome from Australia. Today I'd like to speak about the engagement of active dendritic integration in some forms of circuit computation. We're all, I think, interested in how the brain works and how computations are executed in central networks. And one could imagine that you can view this at different levels, from the synaptic level to the single cell synaptic integration level, leading up to microcircuits and distributed networks that ultimately underlie behavior. And we've spent now decades of our life trying to pull apart how single neurons process information and how those processing schemes are utilized within neuronal circuits to implement defined computations. Now, obviously, the implementation of defined computations is the big problem. So we started in the neocortex of rodents, exploring how electrical signals, EPSPs and IPSPs, spread throughout the dendritic arbor of neurons from their sites of generation down to the site of action potential initiation in the proximal axon. And one of the revelations of this work, together with others, was that neurons are actually really highly distributed electrically and exhibit compartmentalized integrative features, such as in a layer 5 pyramidal neuron. If we challenge a neuron with a burst of excitatory input at the level of the soma, we can generate action potential firing, shown here with the black trace. And those action potentials can decrementally propagate back into the dendritic arbor to these two dendritic recording electrodes. If, in contrast, we give the same input at a distal apical dendritic location, we see a reversal of the pattern of activity, where a dendritic spike is first initiated. And then that forward propagates through the dendritic arbor down to the initial segment to generate action potential firing. And we and others have elaborated these dendritic integration schemes, finding pockets of active dendritic integration, basically in every dendrite that we've looked in layer 5 pyramidal neurons. And the big challenge has been to place this within a circuit computational framework. And together with Jeff McGee, we managed to do that in the rodent neocortex, finding that active dendritic integration was powerfully engaged during an active sensing task. But this sort of is unsatisfactory in that, well, we have to use calcium imaging in order to serve as a proxy of reading out dendritic activity, which is not ideal. So if we want to sort of get to the point of Mars view, tri-level view of how to understand a complex system, then we really have to try a different preparation. And when Ben Sivyer joined our lab, he realized that the answer was right in front of our face, so to speak. It was the retina. The retina implements defined computations. We can define the light stimuli that we shine on the retina. We can remove the retina as a whole neuronal network from the back of the eye and maintain it in ex vivo recording conditions where we have access to the soma and dendrites, and give it natural stimuli. So basically the neural network does its computation, and we can read out from the retinal ganglion cell level, the output level, the pattern of activity generated. The retina has been very well studied, and one of the best studied computations in the retina is the computation of direction selectivity. Direction selectivity, first described by Horace Barlow and colleagues, is a rather beautiful behavior whereby if a light stimuli is moved across the retina in one direction, a train of action potential firing is generated in the output retinal ganglion cell. If the same light stimuli is moved in the opposite direction, basically no action potential response is generated. Now, there's been extensive study of retinal ganglion cells and the circuitry of the direction selective circuitry of the retina at the ultrastructural level, but really no high resolution recordings at an electrophysiological level to try and find out how this computation is executed or implemented in the dendrites of retinal ganglion cells. So we tend to try and do that. And one obvious thing that emerges from this is if we move a preferred direction light stimuli across the receptive field and hence dendritic field of a, in this case, an on direction selective ganglion cell, we see that action potential firing occurs really immediately as the light enters the dendritic arbor. Now, you wouldn't predict that in a passive system because of electrotonic attenuation, i.e. the amplitude of the event at the site of generation, say way distal out here in the dendritic arbor, dramatically attenuates as it spreads down to the soma and the site of action potential initiation, where the axon arises from the soma of these neurons in common with many other types of neurons. So this already emphasizes that something special is happening in this neuron. And what Ben did when he joined the lab was to make simultaneous somatic and dendritic recordings from these on-DS cells in the intact and active retinal circuit. They have really nice direction selectivity, powerfully responding to preferred direction light stimuli, and really not responding at all when the stimuli is moved in the null direction. And what we saw quite spectacularly is as light enters the dendritic arbor, we can record action potential firing from the soma and also what is occurring in the dendrites with a dendritic recording electrode. And what we find is as light enters the dendritic arbor or the receptive field, we see these dendritic spikes initiated. This thing keeps popping up, I'm not quite sure. There you go. Dendritic spikes initiated that forward propagate to the soma and axon to discharge an action potential. As light travels further across the dendritic arbor, we see more and more dendritic spikes generated that drive action potential firing until the light enters the contralateral dendritic arbor, where we see that the action potential firing is occurring at the soma, and then action potentials back propagate into the dendritic arbor. So this tells us that dendritic spike initiation is underlying these wide field characteristics of direction selective ganglion cells. Indeed, if we put very small light stimuli within the dendritic arbor, we can evoke dendritic spikes very readily. And indeed, if we depolarize the dendrite with steps of positive current, we can evoke dendritic spikes. Or if we put the same steps of positive current into the soma, we can evoke action potential firing that then back propagates to the dendritic recording site. So together, this tells us that the dendrites of on-DS cells are incredibly electrically active. And through the production of dendritic spikes, perhaps the wide field characteristics of these cells emerge. And indeed, that's precisely the case. So the retina is fantastic in that TTX sensitive sodium channels are pretty much only expressed in the output neurons of the retinal system. So we can square TTX onto the top of the preparation very locally and know that it's only going to affect the ganglion cells. So we can then use that as a test to see if light stimuli or the engagement of active dendritic integration by light stimuli is necessary for generating an output of direction selective ganglion cells. So this is precisely what Ben did. Preferred direction light stimuli here is sweeping through the dendritic arbor of a DS cell. Is recorded from the soma, the dendrite, and puff TTX locally within the dendritic arbor. Here are the action potential firing evoked under control conditions, and here following local application of TTX. Clearly within the area of TTX application, all the action potential firing has been generated because of the blockade of dendritic spikes. And that's shown very clearly here. If we inject positive current steps through the dendritic recording electrode, we can generate a dendritic spike that precedes and drives action potential firing under control. And then we locally apply TTX and we can completely block dendritic spike initiation. So active dendritic integration is essential for the light driving of action potential output on DS cells of at least the rabbit retina. Wrong way. But what about the characteristics of dendritic spikes? Well, actually we found them to come in two general flavors. These large amplitude dendritic spikes that precede and drive action potential firing, and small amplitude dendritic spikes that seem to have negligible impact at the level of the soma. So we wondered perhaps whether the computation in this neuron was quite interesting in that perhaps these small amplitude dendritic spikes represent spikes generated in small terminal dendrites of on-DS cells. And once evoked, they can propagate or spread out and evoke a dendritic spike in the main parent dendrite. I should mention that these terminal dendrites are the site where most of the excitatory and inhibitory synaptic connections impinge on DS cells. If we look at the light evoked dendritic spike in great detail, what we can find is that it tends to be characterized by two components, a small early component and a larger secondary component that's shown very nicely if we calculate the first derivative of the dendritic recorded spike. This is in stark contrast to the spiking pattern evoked if we inject current directly through the dendritic recording electrode, where we find this simple monorising phase dendritic spike. So this gives us credence perhaps that during light stimuli, there's a dendritic spike generated in terminal dendrites that in turn spreads to parent dendrites, initiating a larger dendritic spike that in turn spreads or propagates to the level of the soma and the initial segment to generate action potential output. And we can sort of test that general idea by blocking sodium channels very locally in the terminal dendrites. And if we do that and we can make a recording distal enough in the dendritic arbor, we can see very isolated terminal dendritic spikes that are completely blocked by TTX in a reversible manner. That's shown clearly here. And in these particular recordings, TTX application at the level of terminal dendrites had no effect on dendritic spike initiation in the parent dendrite, indicating it was completely localized to the level of terminal dendrites. Okay, so now we have this very nice scenario where active dendritic integration in on-DS cells basically reads out the excitatory input coming into the cell. But what about the direction selective mechanism? Well, we think that this multi-site cascade integration scheme that we described for on-DS cells absolutely perfectly allows us to explain the DS mechanism, whereby preferred direction light stimuli give very robust wide field action potential output, and null direction light stimuli give very poor or no action potential output. Now, it's been known for many years that a class of axonless interneuron in the retina, in the output layer of the retina, called starburst amacrine cells, are absolutely essential for generating direction tuning. If you knock these out in many ways, you completely eliminate, or certainly dramatically change, the properties of direction tuning in these output neurons. This is mediated through GABAergic inhibition stemming from the starburst amacrine cells onto the DS cells. And we can make paired recordings between DS cells and starburst amacrine cells and show that activation of starburst amacrine cells produces profound synaptic inhibition. One thing that's also of note is that starburst amacrine cells not only release GABA to causing this inhibition, but they also release acetylcholine, causing excitation. We've recently mapped out in the receptive field, if you wish, of on-DS cells, that starburst amacrine cells placed within the preferred side, that's the side activated very strongly as light moves down to generate action potential output, produce net excitation. Whereas if we activate starburst amacrine cells on the null side, they tend to produce net inhibition. And that net inhibition is all dendritic, in that if we record null direction light evoked synaptic inhibition, we see that it's very large in amplitude at the level of the dendritic recording electrode made in the null subfield, and very small at the level of the soma. So it looks like all the inhibition is acting locally in the dendritic arbor. And obviously, if we block GABA-A receptors in this network, then all the direction selectivity disappears. So we can use gabazine, for example, as a tool to completely prevent direction selectivity. So how does null direction inhibition work within this cascade of active dendritic integration that we see in the dendritic arbor of on-DS cells? So to test that general idea, we drove null direction inhibition by driving a null direction light stimuli that sweep through the dendritic arbor. Remember, that doesn't generate action potential firing. And we could then probe at the level of dendritic excitability by putting current steps through a dendritic recording electrode. If we did that under control and during null direction inhibition, amazingly, we saw absolutely no difference. It's as if the null direction inhibition wasn't there. So the inhibition mediated through the starburst amacrine cell network is not strong enough to overcome the conductances underlying the regenerative generation of dendritic spikes. That left us with the idea, therefore, that perhaps everything was going on at the level of terminal dendrites. And to test that idea, again, we made dendritic recording from the null side of an on-DS cell. And under control conditions, we got very sparse action potential output. And then we locally puffed gabazine within the dendritic arbor, and we could reveal an area of activity as the light traverses over this area where GABA-A receptors have been blocked. And then we interrogated the dendritic spiking activity generated by the null direction light stimuli under those conditions. And we found it composed of both terminal dendritic spikes and terminal dendritic spikes that preceded full-blown dendritic spikes, which in turn preceded action potential firing. If we looked at the amplitude distribution of dendritic spikes, we could see both small amplitude terminal dendritic spikes and large amplitude dendritic spikes. So this left us with the idea that, I'll just cut through this for time, that there's a multi-site integration scheme within on-DS cells featuring terminal dendritic spike initiation, which is like the fuse paper, the fuse that lights a bomb. So if they go off, they can spread into the large primary dendrites where large amplitude dendritic spikes are initiated. Once that large amplitude dendritic spike is initiated, it always propagates to the soma to generate action potential output. So if you can snuff out that bomb, if you wish, by diffusing the fuse, you would prevent light-evoked action potential output. And that's indeed how we think star-based amacrine cell-mediated inhibition is working, by preventing terminal dendritic spike initiation, which prevents this forward cascade of regenerative activity through this highly active dendritic arbor. Okay, so all this work has been based on the rabbit retina. Now, most work, both morphologically and electrophysiologically, has more recently been made in the rodent retina, where there's ideas that the rabbit and rodents differ in terms of their wiring specificity, and indeed other people have suggested that dendritic spiking has absolutely nothing to do with direction selectivity in, for example, the murine retina. So we wanted to test that idea, at least in the rat retina. So what we did was to see if this multi-site integration scheme is preserved across species. We retrofilled identified on-DS cells by putting retrobeads into their brain target and letting them spread back to the retina. Then we could flat mount the retina, image using 2-photon microscopy on DS cells, and then go in, record from them, fill them, and look at their electrophysiological properties. And indeed, if we do that, we find 1, they're on-DS cells, 2, they're highly directionally tuned, and 3, they have 2 cardinal axes as shown in the murine retina. So the $100 question, I guess, is whether active dendritic integration is expressed in rodent on-DS cells, and indeed it seems to be very similar to the rabbit retina. If we make recordings from distal dendritic sites and inject current through a dendritic recording electrode, we can evoke very powerful dendritic spikes that seem to drive action potential output in DS cells. So it seems like across species, at least across rodent-like species, this active dendritic integration mechanism is preserved. In the last few minutes, what I'd like to talk about is unpublished work now about different types of retinal ganglion cells. All our work to date has been based in the on layer of the retina. Obviously, there's the off layer of the retina, and the off layer is sort of super important, right? If you consider, if you're a rabbit, say, what you want to do is not get killed like this rabbit is by this red-tailed hawk. Now, hawks obviously strike during the day. If you're looking up at them into the sky, they appear as a dark shadow. So that's a dark signal on a bright background, which would be ideal for generating activity in off retinal ganglion cells. So we wanted to inquire whether 1, active dendritic integration was manifest in the off layer of the retina, and 2, whether it had any role in other motion computations. And one really startling motion computation first observed by Marcus Meister and published some years ago now in Nature is the idea of anticipatory coding. He recorded 1 class of off retinal ganglion cell and flashed a light. He generated an off action potential firing pattern, both in the salamander and the rabbit. And then he moved a light stimuli across the retina and found, in terms of timing, that these cells demonstrated a sort of anticipatory behavior. Instead of occurring after the offset of the stimuli, they tended to precede the stimuli relative to the flash. So one, they're motion sensitive, and two, they seem to demonstrate some forms of anticipatory coding. So we were intrigued by that. So we looked in the rabbit retina, the off layer of the rabbit retina, and in common with many other groups, have observed two general classes of off ganglion cells, so-called sustained off ganglion cells and transient off ganglion cells. So in response to a light spot, a flashed light spot, sustained ganglion cells show this rather, as the name would suggest, sustained pattern of action potential firing, whereas transient ganglion cells just show an initial very high frequency burst of action potential firing at the onset of this dark light stimuli. What we did observe, however, that sustained cells and transient cells are completely different if we move stimuli through the dendritic arbor. So if we move dark light stimuli across the retina through the receptive field of sustained off ganglion cells, they produce a very lovely pattern of action potential firing when the stimuli is moving at this speed. The transient off ganglion cells, in contrast, are completely disengaged. They're just not interested in such moving light stimuli. And indeed, only when the speed of the stimuli gets faster and faster are transient off ganglion cells engaged. So that leads us to the idea that sustained off retinal ganglion cells encode motion, whereas transient off ganglion cells encode something else. And in support of this idea, the action potential firing rate of sustained off ganglion cells is related to the speed of motion, and their instantaneous frequency distribution of action potential firing basically shifts in a parallel manner as the speed of the stimuli increases. Interestingly also, these cells are incredibly wide field in that their receptive field characteristics do not change as the speed of a moving dark bar is increased. Also, the receptive field is highly correlated with the dendritic diameter, indicating they seem to fire as soon as a light stimuli enters the receptive field. And that's rather shown here, where the time zero represents the time as light enters the dendritic tree, and the sustained cells basically start firing as soon as light stimuli enters the dendritic tree, whereas transient cells either don't fire or fire very late as the stimuli moves across their rather more elaborated dendritic arbors. In contrast to on DS cells, off alpha ganglion cells are not directionally tuned. They show very robust patterns of action potential output to moving light bars, basically projected in any direction across their receptive field. So they have this very circular pattern of action potential output as light stimuli are moved in different directions. They also do exactly what Marcus Meister demonstrated. They show this anticipatory behavior relative to a flash stimuli. So how is that implemented? Well, one thing that we immediately did was just to hyperpolarize the cell and then look to see what lies beneath action potential threshold. And to our surprise, what we found was, instead of a wave of excitation, what we saw was spikelets, thousands of spikelets. And the receptive field of those spikelets was almost identical to the receptive field of the neuron. So it seems like active dendritic integration is engaged in the dendritic arbor of off ganglion cells. And we can show that very clearly by making simultaneous somatic and dendritic recording and injecting steps of positive current through the dendritic recording electrode. And in the sustained off ganglion cell, we see dendritic spikes powerfully forward propagate and discharge action potential firing, whereas in the sustained ganglion, the transient off ganglion cell, we see a completely different pattern. We see basically passive dendritic arbors where action potentials very, very poorly backpropagate into the dendritic arbor. And when excitatory input is given to the dendrite, we see action potential initiation first at the soma and then very weak back propagation. So these cells that are sitting next to each other are responsive both to off stimuli, are night and day different in terms of their dendritic excitability. The sustained cells obviously are dendritic spiking. Those dendritic spikes using the local TTX application method are very similar to on-DS cells. And they're extremely responsive to light stimuli. So if we put a flash light stimuli in the dendrite, we can discharge dendritic spike initiation that drives action potential firing. In the transient cells, if we do the same, we get very powerful dendritic excitatory input, but absolutely no dendritic spike generation, and action potentials are always generated at the level of the soma. If we look at moving light bars, we can see that sustained cells respond right as the light enters the dendritic arbor, whereas transient cells respond at later times. And then if we look using the same techniques as on-DS cells at moving light stimuli, we see dendritic spikes are very powerfully initiated in sustained off ganglion cells, but absolutely not in transient ganglion cells. So last minute, what does this mean to the detection of prey, to light responsiveness in general in the retina? Well, most classes of retinal ganglion cell tile across the retinal surface. That's shown very nicely here for on and off alpha ganglion cells that show this beautiful tiling where the dendrites seem to intermingle just at their ends. So we wondered, therefore, if this exquisite light sensitivity mediated by active dendritic integration in sustained alpha ganglion cells, in essence allowed a vector or a moving wave of information to be generated in the in retinal space in response to moving light stimuli. So we recorded nearest neighbor pairs of sustained off ganglion cells and transient off ganglion cells, and basically just moved a dark light stimuli through their receptive fields, finding the as it enters the receptor field of the first cell, this beautiful pattern of action potential firing driven by dendritic spike initiation. And then as it moves into the second, an equally beautiful pattern of action potential firing. But they're commingled, right, to a certain extent. So there's a degree of co-firing. So basically, in response to this light stimuli, there's no break in action potential firing as the light moves across retinal space. You can imagine another one here, another one here. This is in distinct contrast to the transient off ganglion cells that show this clear break in action potential firing, basically because they don't have active dendritic integration. This sort of moving wave of activity is apparent across all the speeds that we've tested. So in summary, therefore, what we think is that active dendritic integration is super important for computations in the output layer of the retina. We think that active dendritic integration is a feature of motion encoding output neurons of the retina. We think it's a cardinal component of the direction selective mechanism, and we think that it mediates anticipatory responses to generate a moving wave of action potential firing across the off sustained off retinal ganglion cell network. Sorry about going over time. Thank you.
Thank you very much. This was an amazing talk. Really excited. So there are some questions in the Q&A, and also like people that can, if you want, you can also submit your questions in the chat or in the Q&A. So the first one is, do other types of dendritic nonlinearities, calcium or NMDA, play a role in the ganglion cells beside the sodium spike? So this is from Judith Makara, and I actually had also a similar question that has to do with these spikes that you showed to us in the beginning. So you have like these small spikes in the thin dendrites in the terminal branches, and then you had like this, at least they look like much bigger in the more proximal ones. So yeah. This is kind of a similar question. Are these also like sodium spikes, or are they like calcium or something else?
So we think those terminal dendritic spikes are sodium spikes. You can imagine that if they're generated in very fine dendritic compartments, as they spread into a low impedance compartment, such as the large dendrite, the parent dendrite, then they're basically going to vanish out. It's like a dendritic spine, if you wish, right? So then the question is, you'd imagine perhaps the local apparent input resistance in the terminal dendrite to be quite high, where perhaps you could get nonlinear interactions such as NMDA spikes. We certainly don't see NMDA spikes. We can see all this behavior if we put APV on the system, AP5 on the system. So we don't think that's necessary. However, other people do, right? In the murine retina, there's very solid data suggesting that NMDA, multiplicative processing mediated by NMDA receptors, is extremely important for the dendritics, the direction selective mechanism. We certainly don't see that in the rabbit retina, or at least in on-DS cells of the rabbit retina. One thing that I sort of glossed over was the idea of co-transmission in the starburst amacrine cells that both release GABA and an excitatory neurotransmitter, acetylcholine. And our data tends to suggest that acetylcholine itself is a positive influence on the generation of terminal dendritic spikes. So if we block all acetylcholine, nicotinic acetylcholine receptors, the whole gain of this system is dramatically reduced. And we can do that in such a way that we can restore release, if you wish, or mimic release, acetylcholine release at small sites in the dendritic arbor. And if we co-align that with light stimuli, we can recrudesce terminal dendritic spike initiation. So we don't think in the rabbit retina, at least, that NMDA electrogenesis is playing a significant role. However, in the murine retina, it may be different. However, we're working on the murine retina at the moment, finding very active dendritic integration. Now, these two things can, of course, go hand in glove, right? You know, as it in other cortical areas, lots of terminal dendrites have the ability to generate sodium spikes. So clearly, they can go hand in glove.
That's actually really relevant with another question I had, at least, is how compartmentalized are these cells? Like,
oh, they are pretty compartmentalized. So they, sorry, I took that slide out. Their D2S attenuation is very severe, right? Now, we haven't been able to record from the terminal dendrites, and we're going to have to, you know, go to voltage imaging. They're just far too small to record from. But in terms of dendrosomatic compartmentalization, they're as compartmentalized as a large layer 5 cell.
So there is another, thank you. So there is another question. Is there a way to quantify the dendritic structure and see if such structure is related to response to the stimuli? So I think this question had to do with the transient versus sustained cells. So is there a specific dendritic structure for the...
Yeah, that's a sort of interesting question. So yeah, I mean, we can map out whether the dendritic structures are asymmetrical to see if that underlies any aspects of their input-output in space behavior. Their dendrites seem to be broadly symmetrical, their output patterns broadly symmetrical. Is there, I guess the question is there any characteristic, morphological characteristic of a cell that's bearing active dendritic integration? I probably don't think, other than to say that these larger off-alpha ganglion cells, which do have, you know, a dendritic arbor that's 1.2 millimeters across, they're really big and have really large dendrites, where you, if, you know, I'm from the cortex, right? You'd expect dendritic spike initiation to be there to sort of overcome this harsh morphology, but they have none. So obviously there's hallmarks, but whether they're defining hallmarks or causative hallmarks, I wouldn't like to guess at.
Okay, I have one question, if nobody else wants to ask something else. Like, it's a little bit more general. So you said, like, for the sustained firing, there are mostly motion detectors, but I might have missed, like, why do you need the transient cells, like, especially with the figure that you saw in your end of your talk, like the transient cell and these gaps of firing between them? How could this be utilized? Like, what could this be a function?
Yeah, look, I think basically the off-alpha ganglion cells, the transient ganglion cells, really aren't, they respond very nicely to flicker stimuli, to wide field flicker stimuli. So they're clearly geared up for encoding transient changes in luminescence, for example. They're not geared up per se to be a motion detector. So I utilize that gap to indicate the difference between something that's motion detecting with active dendritic integration and those cells that are sitting right next to it that aren't. Now, that's not to say that that's the whole story, right? And I've got another talk here. When we change the adaptation state of the retina, things change, right? And we see some quite startling coupling between these transient off-alpha ganglion cells mediated through very long-range interactions that actually then fill in the gap. But I consider that as a form of function attached to basically this massive adaptation-dependent increase in the whole gain of the system. Basically, the system's trying to pool information in order to detect output, detect input.
Yeah, that's very interesting. So there is a lot of clapping in the chat. I don't know whether you can see it. They're saying thanks for the talk. I have to also transmit this information. So thank you very much. It was really interesting. And yeah, looking forward to the next of your talk, next work.
Okay, thanks very much. See you.
Bye-bye.